Okay, let's unpack this. We're diving deep today into one of the most fundamental challenges for plants living on land.
Mm.
Think about something as simple as a strawberry plant, maybe one right there in your garden.
Mm-hmm.
It absolutely needs water for photosynthesis, its way of making energy from sunlight.
Right, it's essential.
But that very act of opening up its leaves to grab carbon dioxide from the air means it's also losing huge amounts of water.
Yeah, massive amounts. We're talking about something like, 97% of the water the roots soak up just evaporates from the leaves? It's staggering.
It's this incredible balancing act, right? Plants open tiny pores, stomata, to let CO2 in, which they need for food.
But water vapor escapes through those exact same doors.
Yeah.
And the trade-off is pretty brutal.
How brutal.
Well, the figures suggest for every single molecule of CO2 they manage to gain, they can lose hundreds, maybe up to 400, water molecules. Yeah. This constant threat of drying out, while utterly depending on water, has been the driving force behind so much of plant evolution, shaping everything from their roots to their leaves for millions of years.
And a massive piece of how plants manage this challenge boils down to something really unique compared to us, or, animal cells.
The cell wall.
Exactly, the cell wall. This rigid layer outside the cell membrane changes everything about how water interacts with the plant cell.
It really does. It allows them to build up significant internal pressure, which we call turgor pressure.
Turgor pressure. That sounds important.
Oh, it's absolutely central to plant life. It's that internal push against the cell wall, like inflating a balloon inside a box, sort of.
Okay.
This pressure is what makes plant cells enlarge when they grow. It controls whether those crucial stomatopores are open or closed. It helps drive transport systems like moving sugars around.
And it keeps things standing up, like leaves.
Exactly. It's what keeps non-woody parts of the plant, like those strawberry leaves you mentioned, stiff and upright. When they lose turgor, well, they wilt.
Right. Okay, so our mission in this deep dive, pulling from a comprehensive chapter exploring this topic, is to really get to grips with this hidden world of plant water management.
Yeah, how do they actually do it? Taking up water, moving it, losing it.
We'll explore the fascinating properties of water itself, the physical forces that push and pull it through the plant, how individual cells cope with changes in water.
And connect it all back to the plants you actually encounter every day.
Think of it as understanding the secret life of water within a plant.
Mm-hmm.
So let's start with that fundamental truth. Even though water covers most of the earth, it's often the single biggest factor limiting plant growth.
You see evidence of this everywhere, don't you? Look at agriculture.
Irrigation, yeah.
Why is irrigation so critical in so many places? Because even with fertile soil and plenty of sun, if there isn't enough water, crop yields plummet. The source material had diagrams showing, grain yield for barley and wheat having a direct relationship with water availability.
And it's not just agriculture, right? It shapes whole ecosystems.
Absolutely. Water availability shapes entire landscapes. The amount of rain an area gets dictates whether you see lush forests or grasslands or arid deserts. Water literally determines the type of vegetation that can survive and thrive there.
Which brings us back to that paradox we started with. Plants use vast amounts of water.
That 97% loss of transpiration figure is just mind-boggling. Only a tiny fraction, maybe 2 or 3%, is actually incorporated into the plant's tissues for growth or used directly in biochemical reactions like photosynthesis.
It seems incredibly wasteful on the surface, losing so much water just to breathe in CO2.
But it does seem that way. But it's the evolutionary compromise for doing photosynthesis in the air, on land. The pathway for CO2 uptake is physically linked, to the pathway for water vapor loss.
Through the stomata.
Stomata. And the physical gradient driving water out is usually much, much steeper than the gradient driving CO2 in. That ratio, using maybe 400 water molecules for one CO2 molecule, really highlights the intense pressure plants are under to manage water loss.
And effectively managing water relies completely on the unique, almost magical properties of water itself.
It all starts with a molecule, HO. Seems simple, 2 hydrogens, 1 oxygen.
But the oxygen atom is, what, greedier for electrons?
Exactly. It's more electronegative. It pulls the shared electrons closer, creating a slight negative charge on the oxygen side and slight positive charges on the hydrogen sides. This uneven distribution of charge, this polarity, is the key to everything.
And these partial charges mean water molecules act like tiny magnets.
Pretty much. The positive hydrogen end of one water molecule is attracted to the negative oxygen end of a neighbor. These attractions are called hydrogen bonds.
Ah, hydrogen bonds. I remember those.
And while a single one is relatively weak, collectively, when you have billions of them, They are incredibly powerful.
They are. And those hydrogen bonds are responsible for so many properties that are crucial for plants, like water being an excellent solvent.
Definitely. Because it's polar and forms hydrogen bonds, water can effectively surround and pull apart a huge variety of substances, from simple salts to complex things like sugars and proteins. The water molecules interact with the charged or polar parts of other molecules, dissolving them, keeping them dispersed.
And its thermal properties are also linked to those bonds,
Absolutely. Water has a really high specific heat capacity.
meaning it takes a lot of energy to heat it up.
Right. Much of the energy you add goes into breaking or stretching those hydrogen bonds before the water molecules themselves start moving faster and actually getting hotter. This is great for plants because the water in their tissues helps buffer them against rapid temperature swings.
Okay. And it also takes a lot of energy to turn liquid water into vapor.
Yes, that's the high latent heat of vaporization, also linked to hydrogen bonds needing to be broken.
And that's directly relevant to the cooling effect of transpiration.
Precisely. As water evaporates from the leaf surface, it pulls that energy, that heat, from the leaf itself. It's basically the plant's evaporative cooling system, preventing the leaves from overheating, especially under intense sunlight.
Hydrogen bonds are also behind cohesion, adhesion, and surface tension, right?
Spot on. Cohesion is water sticking to itself through those hydrogen bonds. Adhesion is water sticking to other surfaces that it can form hydrogen bonds with.
Like the cellulose in plant cell walls or glass.
Exactly, like cell walls or glass. And surface tension is essentially the energy required to stretch or increase the area of the water's surface where it meets air.
How does that work?
Well, water molecules at that surface are pulled inwards more strongly by their neighbors below and to the side than by the air molecules above. This creates an inward force, kind of like a skin, especially noticeable on curved water surfaces.
And these properties working together explain capillarity, why water can creep up inside narrow tubes.
Yes. Adhesion pulls the water molecules nearest the tube wall upwards. Cohesion holds the entire water column together like a chain, and the surface tension on the curved water surface, the meniscus, creates an upward force on the whole column.
So the narrower the tube.
The higher these forces can lift the water against gravity. This is fundamentally important for how water starts its journey up the plant in very narrow spaces, like within the cell walls or tiny xylem vessels.
And then there's the property that always surprises me. Water's high tensile strength. It resists being pulled apart.
It really does, and again, it's thanks to the hydrogen bonds holding the column together. Imagine pulling on a syringe filled with water. You create tension, or negative pressure, inside that water column.
Probably stretching it.
Kind of, yeah. And the water resists breaking. This ability to withstand negative pressure is absolutely crucial for water transport over long distances in the plant's xylem, though we know from experience air bubbles can get in and break that column.
That's cavitation, right?
Exactly, cavitation, which is a whole other deep dive topic. But the potential for water to sustain tension is vital. We see pressure units like megapascals, MPa, used to quantify these forces.
Give us some context for MPa.
Sure. A car tire might be inflated to maybe 0.2 MPa of positive pressure. Negative pressures, tension in plant xylem can be much, much lower, like -1 MPa or even more negative. It's significant force.
Oh, okay. So with these amazing properties of water in mind, how does it actually get into and move within plant cells? That brings us to diffusion and osmosis.
Right. Diffusion is just the natural random jiggling of molecules because of their thermal energy. Brownian motion.
And it leads to movement from high concentration to low.
On average, yes. It leads to a net movement from an area where a substance is highly concentrated to where it's less concentrated until it's evenly spread out. Basic physics.
But you emphasized earlier, or the material did, that diffusion is only really effective over tiny distances.
That's the critical part for plants, yes. Diffusion is fast over short distances, like a sugar molecule moving across a single cell, maybe tens of micrometers. That could take seconds.
Okay, fast enough.
But trying to move that same sugar molecule by diffusion alone, even, say, a meter, from a leaf to a root, we're talking years, literally.
Years, wow, okay.
Yeah. This is why plants need bulk flow and other mechanisms for any kind of long-distance transport. Diffusion just doesn't cut it.
And osmosis, that's water's version of diffusion.
Exactly. Osmosis is specifically the net movement of water across a selectively permeable membrane, like the plant cell's plasma membrane.
Selectively permeable, meaning it lets water through, but not everything else.
Right. It's driven by differences in solute concentration across that membrane. Water tends to move from a region where its concentration is higher, meaning there are fewer dissolved solutes.
To a region where its concentration is lower, meaning more dissolved solutes.
Precisely. It's almost like the water is trying to spread out and dilute the solutes, increasing the overall disorder or entropy of the system. It's a spontaneous process driven by thermodynamics.
And this is where the plant cell wall changes the game compared to an animal cell.
Completely. If you put a typical animal cell, like a red blood cell, into pure water, water rushes in by osmosis because the water concentration outside is much higher than inside.
And pop.
Pop. Yeah, since there's no rigid wall, the cell membrane just expands until it ruptures. A plant cell, however, has that strong, albeit somewhat elastic, cell wall.
So water still rushes in.
Water still rushes in by osmosis, following the concentration gradient. But as the plasma membrane expands, it pushes against the wall. The wall resists this expansion, it pushes back.
Creating pressure inside.
Exactly. Building up that internal hydrostatic pressure. We talked about turgor pressure. The term osmosis even comes from a Greek word meaning pushing, which fits this pressure buildup perfectly.
Okay, so to really understand why water moves in a particular direction in a plant, we need a concept that combines the effects of both solutes and pressure.
And that concept is water potential.
Water potential, symbolized by the Greek letter psi.
Yes. Yeah, this is the master concept here. Think of it as the potential energy status of water per unit volume. Kind of like water's tendency or desire to move from one place to another.
And it's measured in pressure units? Like MPa?
Usually, yes. Yeah. Megapascals. The fundamental rule is beautifully simple. Water always moves spontaneously from a region of higher water potential to a region of lower water potential.
Higher means less negative, closer to zero.
Exactly. Higher potential means more free energy. Lower potential means less free energy, usually more negative. By convention, pure water at standard atmospheric pressure and temperature is defined as having A of 0 MPa. Everything else, typically in a plant, will be 0 or negative.
And this total water potential in a plant cell is a combination of different factors.
Yes, you can break it down. The total water potential is primarily the sum of two main components: solute potential and pressure potential.
So, S plus EP.
That's the main equation for cell level stuff, yeah. There's also gravitational potential, which depends on height. But for movement within a single cell or between adjacent cells, the height differences are so tiny that gravity's effect is usually negligible compared to the effects of solutes and pressure. We mostly ignore it at this scale.
Okay, so let's break down S and EP. Solute potential, S, relates to dissolved stuff.
Correct. Dissolved solutes reduce the water potential. They make as a negative value, or 0 if there are no solutes, like in pure water.
Why negative?
Because solutes essentially reduce the free energy of the water. They interact with water molecules, making them less free to move or do work compared to pure water. The more solutes you dissolve, the more negative the S becomes.
Got it. And pressure potential, EP?
That's the physical hydrostatic pressure component. Positive pressure, like the turgor pressure inside a plant cell pushing out against the wall, increases the water potential, making it positive.
And negative pressure?
Negative pressure, or tension, which exists in the water column, moving up the xylem, decreases the water potential, making EP negative.
So water moves from where it's happier, higher, less negative, to where it's less happy, lower, more negative.
That's a good way to think about it conceptually. It's always following that free energy gradient, moving downhill in terms of potential.
And this framework allows us to predict which way water will move if we know the water potentials.
Exactly. And these potentials aren't just theoretical. We can actually measure them using various techniques like pressure chambers or psychrometers. It's quantifiable. Oh, and there's also something called matrix potential. Yeah, EP. It accounts for water binding tightly to surfaces, like soil particles or cell walls themselves, especially in very dry conditions like soils or seeds. It's another negative component, important in those specific contexts. But for typical hydrated cells, S and EP are the main players.
Okay. Let's look at how this plays out with a plant cell. The source material had some great examples, like in figures 3.9 and 3.0. You mentioned plant cells typically have a F that is 0 or negative.
Right. So imagine a cell that's a bit floppy, maybe lost some water. We call it flaccid. Let's say its total water potential is maybe 0.7 MPa.
And that's mostly because of solutes inside.
Probably. Let's say its solute potential is 0.7 MPa and its pressure potential is 0 MPa because there's no outward push, no turgor. It's flaccid.
Okay, flaccid cell at minus 0.7 MPa. Now we put it into a solution with a higher water potential, like pure water.
Perfect example. Pure water has MPa of 0 MPa. So you have the cell at negative 0.7 MPa and the surrounding water at 0 MPa.
Water will move from the pure water, higher A, 0 MPa, into the cell, lower, minus 0.7 MPa.
Exactly. Following the gradient. As water enters, the cell volume increases slightly. The plasma membrane pushes against the cell wall.
And turgor pressure builds up. EP starts to increase from 0.
Right. The internal solute potential might become slightly less negative as the cell swells and dilutes its contents a bit. Maybe it goes to negative 0.6 MPa. But the big change is the increase in EP.
And water keeps entering until
the cell's internal total water potential matches the surrounding solution's EP. In this case, the outside is 0 MPa. So water enters until the cell's internal EP is also 0 MPa.
How does it get to 0 if the solutes are still there, making S negative?
Because the pressure potential becomes positive. If S is now minus 0.6 MPa, the cell achieves a total S of 0 MPa by building up a positive turgor pressure of plus 0.6 MPa, because negative 0.6 plus 0.6 equals 0.
Okay, so the inward push from solutes is balanced by the outward push from pressure. The cell is now turgid.
Exactly. Fully turgid in this case.
Now, what if you take a turgid cell, maybe one that's sitting at S equals 0.2 MPa, so it has some positive turgor counteracting negative solutes, and put it into a saltier solution, say one with E equals 1.0 MPa.
Okay, so now the cell's S, n equals 0.2 MPa, is higher than the solution's MS. 1 has 1.0 MPa.
So water moves out of the cell.
Right again. Water will move out of the cell, following the gradient towards the lower water potential in the surrounding solution.
What happens inside the cell then?
As water leaves, the cell volume shrinks. The turgor pressure drops rapidly. The internal solute concentration increases as water leaves, making S more negative.
Could the membrane pull away from the wall?
Yes, if enough water leaves, the plasma membrane might pull away from the cell wall that's called plasmolysis, and the turgor pressure drops, potentially becoming 0, or maybe even slightly negative if the wall pulls inward a bit.
And water keeps leaving until
the cell's internal E equals the solution's, which was minus 1.0 MPa. If the cell loses enough water that its EP drops to 0, it becomes flaccid again, then its entire water potential, minus 1.0 MPa, is determined solely by its now very concentrated solutes, minus 1.0 MPa.
So flaccid or even plasmolyzed?
Depending on how much water was lost, yeah.
The crucial point across all these scenarios seems crystal clear. Water movement across cell membranes is a purely passive process.
Yes, absolutely critical to grasp. There are no metabolic pumps actively pushing water across the membrane against a water potential gradient. That would require energy, and it just doesn't happen for water itself.
It always moves down the water potential gradient.
Always. From higher free energy to lower free energy. The only sort of minor exception is that a tiny amount of water can sometimes be dragged along with solutes that are being actively transported, but the vast majority of water flow, the physiologically significant flow, is purely passive, driven by that efference.
And the properties of the cell itself, especially the cell wall and the membrane, really influence how and maybe how fast this movement happens.
They absolutely do. Take the cell wall's elasticity or stiffness. This affects how turgor pressure changes as the cell volume changes.
How so?
Well, a very stiff wall, which we'd say has a high volumetric elastic modulus, symbolized by epsilon, EP, means that even a small loss of water, and thus a small decrease in cell volume, leads to a large drop in turgor pressure.
So it helps maintain volume, but turgor is sensitive.
Kind of, yeah. It resists volume change as well, but EP drops quickly under stress. Conversely, a more elastic wall with a lower EP allows for larger changes in cell volume before turgor pressure drops significantly. You can hold on to turgor longer as it loses water, but its volume changes more. This relationship is shown in pressure-volume curves.
And this can be adaptive, right? You mentioned the cactus example earlier.
Exactly. It's a brilliant adaptation shown in the diagrams in the source. Cacti stems often have photosynthetic tissue on the outside and specialized water storage tissue deeper inside. The cells in that inner storage tissue often have more flexible walls, lower air, and they can also adjust their internal solutes. During drought, this allows them to lose water more easily, shrinking more significantly than the outer photosynthetic cells.
So they sacrifice their water.
In a way, yes. They sacrifice their own water content, letting their turgor drop, but this helps maintain a higher water potential and thus higher turgor in the vital outer photosynthetic cells, protecting the machinery from making food for longer. It shows how cell wall properties are beautifully tuned to the plant's survival strategy.
Amazing. Okay, beyond the wall, the cell membrane's properties also matter, specifically how permeable it is to water.
Right. That's called the membrane's hydraulic conductivity, often symbolized as LP. The rate of water movement across the membrane depends on two things: the driving force, the water potential difference,
Yeah.
and how easily water can pass through that membrane, LP.
So a bigger difference or a more permeable membrane means faster flow.
Exactly. We used to think water just diffused relatively slowly through the lipid bilayer of the membrane, and that model didn't fully explain the rates of water movement sometimes observed in plant cells. They seem too fast.
And then aquaporins were discovered.
Precisely. Found back in the early 90s. These are protein channels embedded in the plasma membrane and also the vacuolar membrane, the tonoplast. They specifically facilitate the movement of water molecules.
Like little tunnels just for water?
Kinda, yeah.
Yeah.
Highly selective pores that dramatically increase the membrane's permeability to water, its LP value. This makes water flow much, much faster than it would by just trying to squeeze through the lipids alone.
But crucially, they're still just channels. They don't change the direction water wants to move, right? Which is always dictated by the water potential gradient.
Spot on. Aquaporins don't change the driving force or the fundamental direction of water flow from high A to low A. They just increase the rate at which water moves down that existing gradient. They facilitate passive transport.
So they make equilibration faster.
Yes. However, what's really interesting is that plants have found ways to regulate these aquaporins. They can be gated, opened, or closed in response to various signals within the cell, like changes in pH or calcium levels, or even phosphorylation.
So the plant can control how leaky its membranes are to water.
Essentially, yes. This gives the plant another layer of dynamic control over water relations, allowing it to adjust membrane permeability quickly in response to changing environmental conditions like drought or flooding.
So putting it all together, the overall water status of a plant, which we can measure using its water potential, has profound effects on its physiology.
Absolutely. Because of that constant transpiration we talked about, plants growing in natural environments are actually rarely at full hydration, meaning their A is usually significantly below zero.
And when conditions get drier, like during a drought,
Drought causes water deficits, leading to even more negative water potentials throughout the plant. And this inhibits crucial physiological processes. The source material showed a clear hierarchy of sensitivity.
what gets hit first?
Cell expansion, or growth. It absolutely needs positive turgor pressure to stretch the cell walls. So growth is typically the very first process to slow down or stop completely under water stress.
Right, growth stops. Makes sense.
Then, as water potential drops further, processes like cell wall synthesis and protein synthesis start to be inhibited. And eventually, even photosynthesis itself starts to decline as stomata close to conserve water and internal biochemistry is affected.
But this hierarchy isn't just a limitation. It can be part of an adaptive strategy, right?
It can be. For example, inhibiting shoot growth early on conserves water resources. Sometimes, under mild stress, plants might actually allocate more resources to root growth, stimulating the roots to explore deeper soil for water. It's a way plants prioritize resources under stress.
But ultimately, drought does impose absolute limits on life.
Oh, for sure. Despite adaptations, there's a point where water potential becomes too low for survival. Although it's important to note that different plant species have evolved very different tolerances. The specific threshold for critical damage varies widely.
And plants employ strategies like accumulating solutes inside their cells to try and maintain positive turgor under these stressful conditions.
Exactly. That's a key mechanism. By actively accumulating solutes, they make their internal solute potential more negative.
Which lowers their overall internal water potential.
Right, which then allows them to maintain a water potential gradient into the cell, or at least reduce water loss, even when the water potential outside the cell, in the soil or surrounding tissue, is very low. This helps them maintain that vital positive turgor pressure.
We see this in plants adapted to salty environments, halophytes.
Dramatically so in halophytes, yes. But it's also a very common response to general drought stress in many plants. Maintaining at least some turgor is just so critical, not only for growth, but also for keeping tissues rigid and functional.
And how do plants even sense their water status? Is it just turgor?
Turgor is likely a major factor. But there's also fascinating research suggesting other mechanisms, possibly involving stretch-activated ion channels in the plasma membrane. These could potentially sense changes in membrane tension or cell volume as water moves in or out, providing another way for the cell to perceive its hydration level. It's an active area of research.
So to recap this deep dive then, we've journeyed from the fundamental challenge plants face on land needing water for life, but losing most of it.
Through the unique properties of water molecules themselves, those hydrogen bonds doing all the work.
The physics driving water movement via diffusion, osmosis, and that crucial concept of water potential.
To how cell structure, like the cell wall giving turgor and membrane properties influenced by aquaporins, dictates how water moves into and out of cells and how fast.
And finally, we've seen how the plant's overall water status, it's a tie, directly impacts its ability to grow, photosynthesize, and survive alongside the various strategies it employs, like solute accumulation, to cope with water deficits.
Yeah, I think we've covered the core physiological processes, the underlying molecular and physical mechanisms, touched on experimental examples and diagrams that illustrate these concepts, defined the key terms like turgor, S, EP, aquaporins.
And hopefully linked it all back to the real challenges and amazing adaptations of the plants all around us. We really aim to summarize that whole chapter's worth of information.
It's quite incredible when you really stop and think about the constant dynamic interaction with water happening inside every leaf, every stem, every root, all the time.
The precision required to manage those often subtle differences in water potential across different tissues, the elegance of how something as simple as hydrogen bonds underpins everything from maintaining a leaf's rigidity to enabling water to rise dozens of meters in a tall tree. It's complex.
It really makes you look at something as, well, seemingly simple as a wilting houseplant, or the structure of a cactus with a completely new appreciation for the hidden world of water dynamics constantly at play inside.
Absolutely. It's a powerful reminder of the intricate complexity humming away within even the most familiar living things. Perhaps something for you to ponder the next time you're enjoying a cool glass of water. That same substance has such a profound, essential, and secret life inside every single plant.